So Chapter 4, Segment 2 of Behavioral Neuroscience, the textbook, which spans from pages 103 to 111, first starts off by talking about how we can use the word drug in various ways. For example, one definition that one may use is a medicine used in the treatment of a disease, which applies to prescription drugs or over-the-counter drugs, and psychoactive drugs are included in this category, which are compounds that alter the function of the brain and affect conscious experience. They are useful in psychiatric settings and unfortunately may be used recreationally as well and turn into drugs of abuse. And most drugs of interest to behavioral neuroscientists are called receptor ligands. So this first heading of this segment introduces what the next few pages will be talking about, which is the effects of a drug depend on its site of action and dose. So to give an introduction into how receptors and drugs and how they interact work, the text compares them to locks and keys, the drugs being the keys and the molecular receptors being the locks. So since a neurotransmitter can interact with different subtypes of receptors, drugs may be developed to interact with just one or few of a receptor subtype, and selectively activating or blocking specific subtypes of receptors can have varying effects on the body. So when a drug enters the system, the molecules spread throughout the body and bind to receptor molecules that happen to be the correct shape. The molecules don't really actively seek out specific receptor molecules. They just kind of float around and disperse and latch on to whatever receptor they can that happens to fit, like a lock and key, which is the perfect analogy to describe the binding process. And once the drug binds to a receptor, it alters the activity of the receptor, either activating or blocking it. And the binding is usually temporary, and when the drug or transmitter breaks away from the receptor, it returns to its unbound shape and functioning. So on page 104, the textbook gives us a few examples of different neurotransmitters, and there are different subtypes, a few different subtypes of each, and the function of the neurotransmitter in table 4.2. For just to give an example, it gives us the neurotransmitter glutamate and shows that it has various different subtypes like AMPA, kainate, NMDA receptors, and as well as metabotropic glutamate receptors. So the text then goes a little bit further in depth in talking about the drug-receptor interactions and how they may vary in specificity and activity. So a particular drug may bind more strongly to one kind of receptor and less strongly, more weakly to another. A drug that combats anxiety, for example, at low doses, may also produce sedation at higher doses, probably because at higher doses they activate additional types of receptors. So an important term that we should take away from this section is binding affinity, which is the degree of chemical attraction between a ligand and receptor. So a high affinity for a receptor means that the drug will selectively bind to that type of receptor even at low doses and stay bound for a relatively long time, as opposed to lower affinity, which means at that same dose of a high affinity drug, the lower affinity drug will bind to fewer receptors and disattach quickly. So neurotransmitters, surprisingly, have a lower affinity because they bind weakly and thus disattach quickly in order to let the synapse reset for the next signal from the next neurotransmitter. The second major term is efficacy, or the intrinsic activity, the propensity of a ligand to activate the receptor after binding. So agonists have high efficacy, antagonists have low efficacy, and there are also partial agonists, which are drugs that produce a middle-level response. And so the overall action of a drug is determined by the combination of the level of affinity and efficacy. So the next section talks about the dose-response relationships between drugs and how they can reflect the potency and safety of those drugs. So greater doses of drugs lead to greater effects because larger doses increase the proportion of receptors that are bound by the drug. And so a graph has been developed in order to plot the relationship between drug doses and their observed effects. And these graphs are called dose-response curves and serve as one of the main tools for understanding pharmacodynamics, which is the functional relationship between drugs and their targets. So dose-response curves can help reveal a few characteristics of drugs, for example, their effective dose ranges, and they can help compare potencies and efficacies of different drugs side by side, and thus help reveal which drug is safer to use. So the next section talks about tolerance and how repeated treatments from drugs may reduce their effectiveness. So our body systems can change their functioning in order to accommodate environmental challenges, i.e. drugs, and develop a drug tolerance, which is a phenomenon in which a drug's effectiveness diminishes over repeated treatments and the body becomes less responsive to a constant dose. Consequently, larger and larger doses of a drug are needed to cause the same effect as the original dose did. So there are various kinds of tolerance, three main ones that the text talks about. The first one is metabolic tolerance, in which the body's metabolic systems and organs become effective at eliminating a drug from the bloodstream before it has a chance to affect the brain or another target. The second kind is called functional tolerance, in which the target tissue's postsynaptic membranes change the number of receptor proteins on the neuronal membranes. And so repeated exposure to agonists results in the neurons down-regulating or decreasing the number of receptors to which the drug can bind, and thus decreasing sensitivity. Or they may upregulate as a result of repeated exposure to antagonists, and they increase the number of receptors and thus increase sensitivity. The next section of the text talks about drug tolerance and how repeated treatments may reduce the effectiveness of a drug. So our body systems can change their functioning in order to accommodate environmental challenges, i.e. drugs, and develop drug tolerance, where a drug's effectiveness diminishes over repeated treatments and the body becomes less responsive to a constant dose. And so in order to cause the same effect as the first treatment did, you would need larger and larger doses as time goes on. So the three main types of tolerance that the text gives are metabolic tolerance, functional tolerance, and cross-tolerance. The first kind, metabolic tolerance, is where the body's metabolic systems and organs become effective at eliminating a drug from the bloodstream before it even has a chance to affect the brain or another target. The second kind is functional tolerance, where the target tissue's postsynaptic membrane changes the number of receptor proteins on the neuronal membranes. And so when there is repeated exposure to agonists, the neurons downregulate, which means that they decrease the number of receptors to which the drug can bind to, and thus decrease sensitivity. Or when there is repeated exposure to antagonists, the neurons upregulate, which means that they increase the number of receptors and thus increase sensitivity. And the third kind of tolerance is called cross-tolerance, where the development of tolerance for one drug causes tolerance to develop for another drug, often one of the same chemical class. So as a result of drug tolerance, you could stop taking the drugs, and this would result in withdrawal symptoms, which are symptoms arising when one stops taking a drug that they frequently took it, especially at high doses. And another key word to take away from this section is sensitization, which is where some drug responses can become stronger with repeated treatments rather than weaker. And so the body shows an enhanced response to a drug after repeated doses. The next section talks about how drugs are or can be administered and eliminated in many ways. So the amount of drug that reaches the brain and the speed of action are in part determined by the drug's route of administration. A keyword that we should know is bioavailable, which is a substance or drug that is present in the body that is able to interact with physiological mechanisms. So smoking or intravenous injection rapidly increase the concentration that is bioavailable versus oral ingestion and similar routes of administration in which the drug concentration builds up more gradually over time. And the duration of a drug effect is largely determined by the manner in which the drug is metabolized and excreted from the body via either the kidneys, liver, lungs, etc. Another key word that we should take away from this section is biotransformation, which is the process in which enzymes convert a drug into a metabolite that is itself active, possibly in ways that are very different from the actions of the original substance. And so the biotransformation is important because of drugs, they can be a source. So another key word from this section that we should know is biotransformation, which is the process in which enzymes convert a drug into a metabolite that is itself active, possibly in ways that are very different from the actions of the original substance. And so this term is important because the biotransformation of drugs can be a source of unwanted side effects, which are not fun. In addition, pharmacokinetics are the factors that affect the movement of a drug into, through, and out of the body. And the blood-brain barrier is something that we reviewed back in Chapter 3, I think. And it poses a major challenge for drugs that are too large to pass through this barrier and want to enter the brain but cannot. So the next major section that the text introduces is how drugs affect each stage of neural conduction and synaptic transmission. So to give a little intro, it talks about local anesthetics, and which are drugs that block sodium channels, thus action potentials to stop neural transmission and pain fibers. So some examples of these drugs are Procaine, or by the brand name that we know it by, Novocaine, as well as lidocaine. And most drugs that act on the nervous system do so by changing synaptic transmission, either presynaptically or postsynaptically. The text first talks about how they change presynaptic neurons, though, and as a result, the drugs change the system that converts an electrical signal, i.e. an action potential, into a chemical signal, i.e. the secretion of the neurotransmitter. So the most common presynaptic drug effects, either on transmitter production, release, or clearance. So the first one, transmitter production. In this process, the drugs may inhibit enzymes that neurons use to produce a neurotransmitter. They could block the axonal transport, meaning the materials don't reach the axon terminals and the neurotransmitter, as a result, is not produced. Or they can block the ability to store the transmitters into vesicles. The second type, transmitter release, is where certain drugs may prevent the release of a specific neurotransmitter, even if the action potential does reach the terminal. So, for example, calcium channel blockers block the calcium influx that normally drives the release of a neurotransmitter into the synapse. And the main ingredient in Botox, called botulinum toxin, this toxin blocks calcium-dependent release of acetylcholine, which causes muscle paralysis. A major term that we should know from this section that relates to transmitter release is called autoreceptors, which are receptors for the synaptic transmitter on the presynaptic membrane that monitors and tells the axon terminal how much transmitter they have released, acting as a feedback system. And drugs can alter these signals and provide a false feedback system to the axons. And also caffeine blocks autoreceptors of endogenous ligands a specific endogenous ligand called adenosine, which helps to control the synaptic activity by inhibiting neurotransmitter release. And thus, by blocking adenosine, caffeine increases catecholamine release, which translates to us as arousal. And so the third major category is transmitter clearance. And this is where drugs can interfere with transmitter reuptake, which is the reabsorption of synaptic transmitters by the axon terminal from which it was released. And they can do this by blocking transporters, which are the specialized proteins that normally remove the neurotransmitter from the synaptic cleft for reuse. And so antidepressants commonly inhibit reuptake of serotonin, for example, and thus allow it to accumulate and have a larger effect on the postsynaptic receptors. And drugs can also influence and interfere with degradation, which is the chemical breakdown of a neurotransmitter into inactive metabolites. They may inhibit this process and allow for an accumulation of the neurotransmitter, which leads to a greater effect on the postsynaptic cell as well. And so the other way that drugs can affect the nervous system, specifically synaptic transmission, is on postsynaptic cells, which the section briefly introduces before it goes into more depth with the following pages. But just to state the facts, drugs may also modify the activity of the postsynaptic cell through two major actions, either by affecting the transmitter receptors or affecting the cellular process with in the postsynaptic cell.